Okay, so hi everyone. I'm really excited to talk to Beacon about some of the work that I've been doing during the last couple of years as a postdoc. And so as a postdoc, I've been studying the evolution of vision-related genes in jellyfish. And this is something that I've been interested in a long time. So I'm really excited that I get to work on it. Sorry. So a large question that I try to address in my research is how do complex systems evolve and diversify? And I'm particularly interested in the evolution of eyes because these structures are energetically costly, yet we have seen them evolve multiple times independently. Image forming eyes have evolved its thought at least 40 times, but with advances in Evo-Devo, it seems like the number is much larger. And as I mentioned, visual systems are energetically very costly. So animals that are adapted to dark environments, such as underground or caves, have downregulated their eye size. So they have smaller eyes. This is an example of the Mexican blind cavefish. The eye is very small in the cavefish species, and it's under a layer of skin. And here I have a surface beetle and a cave beetle, and the cave adapted beetle has much smaller eye that has also lost pigmentation. And in addition to eye reduction, it has been found that genes associated with phototransduction are also downregulated in cave species. Now, the phototransduction cascade is the one that is responsible for light detection. Photoreceptor cells are light-sensitive cells, usually in eyes, that have opsin proteins in their transmembrane. So when photons of light hit the opsin, it becomes activated, now called metarhodopsin. And this triggers a G cascade, which is followed by enzymatic reaction and second messengers that then cause the ion channels to open or close. And this varies depending on the photoreceptor cell type. And then finally, there is termination of phototransduction by turning off the rhodopsin, having it be inactivated and ready to be activated again when another photon of light hits it. So in animals, we see a diversity of eyes, but the photoreceptor cells are divided into two types due to their structure. We have ciliary photoreceptors, shown here in this image in pink, and rhabdomeric photoreceptors, shown here in blue. The phototransduction cascade has only really been described for Drosophila as a model of R-opsin phototransduction, and in some vertebrates as a model of C-opsin phototransduction. But how representative are these models? So in insects, everyone uses a candidate gene approach using Drosophila phototransduction models in order to do some of their studies. So for my PhD work, what I did was I looked at the model using Drosophila and looked at how these genes are changing in moths and butterflies, which are adapted to different light environments. Here again is a schematic of the phototransduction cascade. And what I found is that there are differences between moths and butterflies to Drosophila that are due to gene gain or loss. So the differences between these two more distantly related species have to do with gene duplications or gene loss along the way. And differences between moths and butterflies were mostly due to differences in differential expression. So moths and butterflies had the same genes, they just had differences in expression, probably due to their adaptation to different light environments. And this is evident when we look at their ion channels, TRP, transient receptor potential ion channel, and TRPL, transient receptor potential like. When I looked at the expression of these genes, the expression here is plotted as the log2 of transcripts per million. In orange is the expression in Manduca sexta, the moth, and in red is the expression in butterflies. And what I found is that TRP is expressed much lower in moths, and this is the ion channel that is thought to function in high light conditions because it's potentially much faster. Now, since I saw that there was differences between closely related species, it brought about the question, what components of the cascade were used for signaling in an ancestral animal? And how did photoreceptor cells diversify, and what structure in the ancestor did these photoreceptor cells come from? And what was the cascade like in an ancestral species? So that brought me to what I'm working on now, which is looking at cnidarians, which are thought to be more primitive and sister to bilaterians. Cnidarians have C opsins that are closely related to bilaterian opsins. So this is a phylogenetic tree of opsins. C opsins and R opsins are the visual opsins. In addition, in cnidarians, eyes have been found to have evolved independently at least 9 times and potentially more. And there's one group of cnidarians, cubozoans, the box jellies, that actually have complex lens eyes. So not much is known about cnidarian phototransduction, but there are some things that we do know from previous studies. So a study in Hydra looked at the expression of an opsin gene and of an ion channel CNG, cyclic nucleotide gated channel. This is in situ staining, and in the merged image, it seems that these two are co-localized. They also looked at opsins and arrestin. Arrestin is a component that terminates phototransduction. And when merged, it looks like these are co-localized as well. So presumably, cnidarian phototransduction is using a CNG ion channel and arrestin to terminate phototransduction. And then another study looking at cubozoans, which have lens eyes, found that opsin expression is co-localized with expression of a G alpha S component. So the phototransduction cascade in cnidarian might be transduced by this G alpha S. And for reference, Drosophila uses G alpha Q, and vertebrates use G alpha I, which I'll come back to in the end. So I was interested to see what light detection was like in Hydra. And I asked the question, how are opsins evolving in Hydra? What are the expression patterns of opsin genes? And what candidate phototransduction cascade genes are found in Hydra? Hydra is this cute little polyp, and it's a good model system because it's been studied for over 270 years. Currently, it's arisen in popularity, and it's being used to study stem cells and multipotency. It's also interesting because it's capable of head regeneration, and it reproduces asexually by budding. And Hydra actually can regrow a full body from just cell aggregates. So it's pretty interesting in terms of the molecular basis of development. We also have a reference genome, which helps a lot when looking at a lot of these studies, and it's easy to maintain in the lab. And one reason that I think Hydra is a good model is because it's sister to bilateria. So I think that studying genes and some of these connections can tell us about evolution or conservation of genes and gene networks. So Hydra do like eyes, and the most simple definition of an eye is a photoreceptive cell next to an eye in a pigment cell. And Hydra do not have that pigment cell, but they do probably have photoreceptive cells because they react to light. So if you shine a light, the Hydra will scrunch up. So what we did was we extracted RNA from tentacles, the hypostome, body column, the budding zone with a little bud coming off, and foot of a Hydra, of Hydra polyps. And then I used Trimomatic to trim and pair RNA-seq reads. Because the reference assembly is not that great, I used star and string tie to make a transcriptome guided assembly. Then I read mapped the reads back to the guided assembly using bowtie and RSEM to quantify reads. And I extracted the transcripts per million. So this was in order to see the expression patterns of opsin genes. And then to see how these opsin genes are molecularly evolving, I took known opsins from other cnidarians from GenBank and did a homolog search of the genome using command line BLAST. But in order to identify potentially missing opsins, I also used BLAST to go to annotate the genome and searched those annotations for opsins or opsin-like genes. And I aligned the genes and generated a phylogenetic tree using Mega. So what we found is, it's shown here, this is an opsin, a cnidarian opsin phylogeny. In color are different cnidarian species, and in black are the outgroups. We were able to recover known groups such as the ciliary and rhabdomeric opsin. So what is really interesting is that based on color, we can assume that these genes are evolving by lineage-specific duplications because the opsins are grouping by species. And in Hydra, we found 45 opsin genes. Now, we named those genes according to their location on the genome. So if you could see this here, D22, D23, and D24 are next to each other on the phylogeny. So that means that the sequences are very similar, and they're next to each other on the genome. So that implies evolution by tandem duplication. So presumably these genes are evolving in Hydra by tandem duplications. And what was new from our study is that I identified 2 genes that group outside of previously defined groups for Hydra. So Group C and Group D were previously described in a 2008 paper that was done using mostly PCR. So now that we have a genome and more data, we were able to find 2 opsin genes that are now grouping together with those that were previously known. So to answer the question, how are opsin genes evolving in Hydra? They are evolving by lineage-specific duplications and potentially by tandem duplications. So next, I wanted to see what the expression patterns of opsin genes are. And here on the x-axis is the list of the genes that we found. This is only some of them, and one of them I will show after. In the size of the circle is the expression in transcripts per million, and the different colors correspond to different tissues. So in terms of the genes that are upregulated in certain tissues, B1, D2, D14, and D37 we found to be more highly expressed in the hypostome. C2, D4, and D24 were more highly expressed in the tentacles. And D36 and D38 were more highly expressed in the foot. We did not find any differentially expressed genes between the body column and the budding zone. But the reason that we were looking at all of these tissues is because it's not known where light detection is happening, and people are not sure if they're detecting light using the foot or the hypostome, which is sometimes referred to as the head, the hypostome together with the tentacles. And another really interesting thing from this paper is that the one of the opsins that we found to group outside that was not previously described, this opsin, which we called Hydra opsin A1, was actually expressed 200-fold more than any other opsin gene. And it wasn't upregulated in any of the tissues, so it was expressed everywhere. And we did not know, the potential function of it. So I looked at some single cell data. As I mentioned, Hydra is like a rising in popularity. So around the time that I was working on this paper, another paper came out where they looked at trajectories of stem cells in Hydra. So, and then they made their atlas publicly available in a genome browser. So I looked for our gene of interest. And I found that the Hydra opsin A1 is predominantly expressed in nematoblast and nematocyte cells from the interstitial cell lineage. So these are the stinging cells. And it was previously found that these stinging cells actually are activated by light. So if you fan light, sometimes you can get the stinging cells to sting, to fire. We also found some expression in the basal disc and battery cells of the ectodermal epithelial cells. So I just want to go back really quick to talk about the opsins and their functions, because for opsins to be visual, a lot of times they require what is called a chromophore molecule, and a lot of times this is 11-cis-retinol. So it's in the 11-cis-retinol configuration, and when the photon shines on it, it turns into all trans, and that's what causes the opsin to become activated. So visual opsins all have a conserved binding site. It's a lysine, and it is what's used to bind the retinol to make it light detecting. But the gene that we found to be highly expressed in a lot of these tissues actually was lacking the conserved lysine. So that made us think that it could potentially have a different role in vision, or it might be used for a different sensory modality. In butterflies, I found an opsin that is used to photoisomerize the chromophore, and opsins have also been known to detect heat or sound. So they're very similar to gustatory and olfactory receptors. So it may be that this what we call an opsin gene is not used for light detection, but might be detecting something different. So in terms of what are the expression patterns of opsin genes, we were able to identify genes that are differentially expressed in different tissues. Some of them are upregulated in the hypostome, some in the tentacles, and some in the foot. So what about candidate phototransduction cascade genes? Which ones are found in Hydra and which ones are expressed in Hydra? So what I did for this was I took sequences for the components of Drosophila transduction and also vertebrate transduction, and I identified homologs of this in Hydra. And then I looked at their expression patterns. So they are shown here. The dark green means they are more lowly expressed in that tissue, and the dark purple means that they are more highly expressed. And here's the tentacle, hypostome, body column, budding zone, and foot, and the name of these genes. So these are all potentially associated with transduction after the opsin gene is activated. And what I found was that a lot of these genes that are homologous to phototransduction cascade components are more highly expressed in tentacles and hypostome. So next, what I wanted to do was look at the expression patterns and clustering of opsin genes together with phototransduction cascade genes. So I put sequences and their expression together, and I generated a heat map and looked at how they cluster. And here, the low expression is dark blue and the high expression is dark red. We have a lot more samples here because we included stages in budding and also different time points in head regeneration. So this allowed us to have more patterns to find increases or decreases in expression. And the genes that cluster together, we still see that phototransduction cascade components cluster together. And we found two opsins that have similar expression patterns to these homologous phototransduction cascade components. That was C5 and D1. And these opsin genes both have conserved retinal binding lysine site. And one thing to note is that since Hydra have C opsins, I expected them to have homologs of vertebrate cascade components because vertebrates have C opsins, and I did not expect them to have R or rhabdomeric components, but we did find that they have that. So here PLC is phospholipase C, and this is used in Drosophila phototransduction. So that was an interesting surprise. So in terms of what candidate phototransduction genes are found in Hydra, I found that homologs of both vertebrate and insect phototransduction cascades are in the Hydra genome, but also being expressed in Hydra and potentially being co-expressed with opsins. So here's the schematic of the phototransduction cascade in cnidarians based on what we know from previous studies, but nothing has been tested functionally, so this is just a model. But I'm able to add to this model because I found an SEC14-like gene, and this is presumably transporting a chromophore to an opsin. But in addition to the known G-alpha S, I also found G-alpha I and G-alpha Q having similar expression patterns to other phototransduction components. And G-alpha I is the G-alpha component used by vertebrates, and this is the component used by Drosophila. Similarly, I found GMP-PDE, which is a phosphodiesterase, and PLC, the phospholipase C. And this one is from vertebrate phototransduction. This one is from the Drosophila model, but somehow these are also being expressed in Hydra and in similar patterns to other components. And in terms of termination, arrestin was known, and I did recover that gene as being co-expressed with phototransduction cascade components, but I also identified a G protein coupled receptor kinase and an RH kinase, which can be used to terminate phototransduction by phosphorylation. So that was like really interesting to me that we were able to find these components in Hydra. But although we can use Hydra to look at the molecular evolution of opsin genes and look at the expression of these genes and phototransduction cascade components, we can't make any definite conclusions based on whether or not these genes are involved in light detection, because Hydra lack a specialized light detection organ or an eye. So we can't really conclude much about what these genes are being used for and what the cascade is like. So that brought me to my next project, which was investigating the light detection in Tripodalia. And Tripodalia is part of Cubozoa, which have complex lens eyes. And here is an image of a rhopalia. So Tripodalia have 4 rhopalia, and these are structures that have the eyes in them. So Tripodalia have 4 rhopalia. Each rhopalia have 1 small upper lens eye, 1 lower lens eye, 2 pit eyes and 2 slit eyes. Tripodalia are monochromatic, which means that they do not have color vision. They don't discriminate between different wavelengths of light, but they do detect blue-green wavelengths. And they have also been known to use vision to navigate towards landmarks. So in Puerto Rico, where they are found, people have displaced them from where they want to live, and they go up to the surface and find the mangroves where they live. And so they're using vision to navigate. Another reason that these are interesting species to look at is because they are different from other medusa in that Tripodalia fertilized eggs develop within the female, and the polyp undergoes metamorphosis into a single medusa. So typical jellyfish medusa, what happens is you have a male and a female, and they spawn, and then the fertilized egg develops into a free-floating planula, and then the planula settles, and you get a polyp, which segments, and then the segment buds off, and you get a juvenile medusa that then grows into an adult. But in Tripodalia, you have internal fertilization, and then the planula is released, and more importantly, the polyp goes directly into creating a juvenile medusa. So here I'm showing some images of Tripodalia. Here, this is a planula. Here is a polyp that is developing its tentacles. Here's an attached polyp. This is a polyp undergoing metamorphosis, and so we can see the rhopalia starting to form here, and the juvenile already has those eye structures. And here is what the lens looks like. This is the staining, and R is the retina, and L is the lens cells. So for Tripodalia, we had similar methods. We took some publicly available RNA-seq from a polyp undergoing metamorphosis, a whole adult, rhopalia, tentacles, manubrium, and body not including these tissues listed above. And I took the RNA-seq data and mapped the reads to a transcriptome shotgun assembly using bowtie and RSEM. And then I extracted the TPM to look at how these genes are being expressed. I used transdecoder to find the coding sequences, and I used NCBI and BLAST again to find candidate genes in those peptide sequences. So for Tripodalia, I asked similar questions, but now I wanted to know what opsins are being expressed in the rhopalia, in this light detecting structure, and what candidate phototransduction cascade genes are co-expressed with these opsins. So in our lab, 2 opsins that are known to be expressed in the rhopalia were studied using in situ hybridization. And what was found is that one of the opsins that we called lens expressing opsin is found in the lens, but the other opsin was actually found outside of the lens. So this was called a neuropil expressing opsin, and it was not associated with the lens eyes or the simple eyes. So when I looked at this using a transcriptome-wide approach, I found that this gene, so here again is the gene ID. These are known Tripodalia opsins, and these are some IDs from our transcriptome. And here is the lens expressing opsin. So the lens expressing opsin was very highly expressed in the rhopalia, in the eye structure. And it had some expression in the whole adult and some expression in the polyp undergoing metamorphosis. But out of all of the opsins, this was the most highly expressed one, and in the tissue where we expected light detection to be occurring. So for light detection in Tripodalia, what we found is that the lens eye opsin is upregulated in the rhopalia. And next, we wanted to see what candidate phototransduction genes are being co-expressed with those opsins. So I did something similar where I took the phototransduction cascade components from Drosophila and also from vertebrates, and I found homologs of these in Tripodalia. And then I looked at the different tissues in the polyp and the polyp undergoing metamorphosis in the Europalia, body, whole adult, tentacles, and manubrium. And the expression here is shown, the low expression is dark blue and high expression is red. Here is the opsin that we were interested in. So we see that it is expressed in the Europalia more so than in other tissues. C and G, which is known to be involved in light detection, actually is also being expressed in the manubrium, which is called sometimes the mouth. And arrestin, another gene that was known for cnidarian phototransduction, is more highly expressed in the Europalia. We see GS to be more expressed in the Europalia. And G-alpha Q, which I mentioned, I found in Hydra to be co-expressed with phototransduction cascade-like genes, was actually more highly expressed in the Europalia. So based on this, there were many genes that are homologous to phototransduction cascade components that are more highly expressed in the Europalia relative to other tissues. So again, we do find homologs of both vertebrate and insect phototransduction cascade components in Cnidaria. And so building off of what we learned from Hydra and Cnidaria, this is what has been previously described. And we can add to it, in Tripodalia, we also find a SEC-like gene that is being upregulated in the Europalia. We find G-alpha I and G-alpha Q also highly expressed in the Europalia. And we again find PDE, but PLC is not more highly expressed in the Europalia. Now we found another gene, adenylate cyclase encoding gene. We again find GRK and RH kinase, so G protein coupled receptor kinase and an RH kinase. And very interesting, we were able to find a homolog of TRP. TRP is the receptor used in highlight conditions in Drosophila. So differences between Tripodalia and Hydra are these two here. So even though they're in the same group, we do find differences in their potential phototransduction cascade. So some of the conclusions from both of these projects and some future directions. We did identify opsins that have similar expression patterns to phototransduction cascade gene homologs. However, we need to test whether or not these genes are orthologs. So I have mentioned that they're homologous, and the reason that I was very careful about my wording is because we need to make phylogenetic trees to see whether or not these genes are orthologs or not. And then in Tripodalia, we found genes that I mentioned are more highly expressed in the Europalia. However, I did this using a candidate gene approach. So we need to confirm their upregulation and co-expression using a genome-wide approach. And this is what we're currently working on. I also mentioned that we found homolog components of R opsin and C opsin expressed in the Europalia, but we need to localize their expression. So we need to see if these genes are, so using in situ or immunohistochemistry, we need to see if these are expressed in the lens or with that opsin that I mentioned was expressed outside of the eye, but in the Europalia. And then the reason that I have been working with jellyfish is that then we can look at how some of these genes are being expressed in independently evolved cnidarian eyes. And what this will tell us is whether or not these convergently evolved eyes have convergence in their gene expression or if they're using different genes. And so back to that big question that I'm interested in, how do complex systems evolve and diversify? When it comes to vision, since we found R opsin and C opsin components in cnidarians, it's possible that these were present before the cnidaria and bilateria split, and they were being used to transduce light, but somehow some of the components were lost and are just fine-tuned in Drosophila now and in vertebrates. Or it's possible that these were present before the split, but used for some other type of sensory modality, and then were later co-opted for vision in independently evolved eyes. So with that, I would like to thank my lab. The Hydra work was done in Ali Mordasavi's lab at UC Irvine, especially with the help of Robbie and Heidi, and a lot of advice from Rob Steele. And the Tripodalia work was done in Todd Oakley's lab, mostly working with a grad student of his, Natasha Pisciani. And then I'd like to thank my funding sources. So these are my 2 fellowships for my postdocs, Georgie Hewitt Foundation and the UCSB Chancellor's Fellowship Program. And I'll take any questions.
Thank you, that was great. I'd like to encourage everyone to ask any questions you might have either by unmuting yourself or by using the chat box. Either is fine. I had no idea there were jellyfish with eyes. This was really fascinating.
Yeah, thank you. I know. Unmuted. Yeah, when I learned that jellyfish had eyes, I was like, I must study phototransduction in these species.
Yeah, very cool.
Thank you.
Yeah, I also didn't realize that opsins were similar to olfactory receptors.
Yeah, so one thing that I'm really interested in doing sort of long term is seeing how they have evolved, because they are so similar. I believe it's possible that they evolved from a similar ancestral receptor.
Cool, very cool.
Yeah, thank you.
So I have a question, actually. So you had mentioned that these cnidarians are perceiving things monochromatically, but they're sensitive really only to the blue-green spectrum. Do you have a sense if organisms from different depths over evolutionary timeframes would be likely to have a different spectral sensitivity because you get different wavelengths penetrating to different depths in the water column?
Yeah, that's a really great question, because it's something that I've been thinking about recently, because Hydra are freshwater and found near the surface, and Tripodalia are also found near the surface, but they're marine, and they both respond to blue wavelengths of light. So I was wondering the same question that you are, and I'm really interested in knowing the answer to that. So the answer is I don't know, but I would like to study that. And then I would assume, similar to you, that at different depths, they're probably responding to different wavelengths of light. Yeah, but I don't know. But I think that's really interesting, and I want to know why that is. Yeah.
Do we have any other questions? It doesn't look like it. Thank you for such an excellent and very clear talk. That was really enjoyable. Thank you.
Yeah, thanks. I'm glad to talk to Beacon about it.
Always happy to see you. So one more round of applause.
Thank
Yay.
All right, well, I guess I will stop the recording now. Nope, I'm not doing that. Not leaving meeting, stopping.